Hello, today I'll tell you about my research where we investigated the role of G alpha 13 in mouse models of pancreatitis and pancreatic cancer. G alpha 13 is a G protein that regulates GPCR signaling. It's been described both as a tumor suppressor and a tumor promoter. Our lab has previously reported that G alpha 13 is upregulated in pancreatic cancer and promotes invasion in vitro. Today, I'll provide an update on our ongoing investigation of GF13 in pancreas cancer using mouse models. We first began by conditionally deleting GF13 in a well-characterized mouse model of pancreas cancer, the KPC mouse model. We investigated the effect of loss of GF13 on tumor initiation at both 3 and 6 months and saw that there was no difference in the overall burden of the tumors. Subsequently, we saw that there was no difference in the overall survival of these mice. We then investigated the effect of GF13 loss in the KPC mouse model. And in this context, we saw that loss of single copy of GF13 results in worse survival of these mice and more advanced tumors. We next investigated the role of GF13 in the cancer cell lines, and by establishing cell lines from these mice, we saw that loss of GF13 results in poor growth in, reduced growth in 2D and 3D environments, which is consistent with previous reports. And when we injected these mice in vivo, we saw that the cells failed to grow in the subcutaneous space. We saw a similar effect when we used shRNA to inducibly knock down GF13. We know that pancreatitis is a risk factor for pancreas cancer, and it has been shown to promote tumor development in mouse models. So we investigated the effect of loss of GF13 in pancreatitis using a cerulean-mediated model. Here we saw that loss of GF13 results in more tissue injury, more release of serum amylase. We saw in the chronic pancreatitis that there is more acinar ductal metaplasia. And also overall, we see increase in inflammation where we see elevated inflammatory cytokines and increased infiltration of immune cells into the tissue. In summary, we found that loss of GF13 worsens survival in KPC mice, but when we established these cell lines and implanted them in syngeneic hosts, that they failed to grow. We observed that loss of GF13 exacerbates tissue injury and inflammation, and we demonstrate that GF13 can have opposing function in tumor biology depending on the context and modeling approach. In the future, we will examine whether the inflammatory microenvironment may dictate the opposing function of GF13 in pancreas cancer.